The observation deck of the Quantum Horizon Research Station hummed with the subtle vibrations of advanced technology. Dr. Zarakil adjusted the prismatic lenses of her analysis interface, her manipulative tendrils moving with practiced precision across the holographic displays. Another standard rotation, another batch of biological samples to categorize and file away. The human specimens had arrived 3 solar cycles ago, packaged in standard diplomatic containment units that bore the official seal of the Interstellar Research Consortium. Most xenobiologists would have found the assignment mundane, but Zarakil prided herself on her meticulous approach. Every sample, no matter how seemingly insignificant, deserved comprehensive investigation. She pulled up the initial diplomatic reports, her optical sensors scanning through the digital documentation. Humans, a relatively young species from a small blue-green planet on the outer rim of the Milky Way galaxy. Their technological development was considered rudimentary by galactic standards: chemical-based combustion engines, primitive quantum computing, and biological systems that appeared woefully unmodified by genetic engineering. The preliminary reports painted a picture of a species that was more potential than achievement. They had only recently achieved interplanetary travel, with their first tentative steps beyond their home system occurring less than a standard century ago. Their biological records suggested a species riddled with vulnerabilities, susceptible to environmental changes, prone to genetic mutations, and lacking the sophisticated bio-augmentation common among advanced civilizations. Zarakil's research assistant, a young neural-linked technician named Vox, approached with additional documentation. The diplomatic team was quite impressed with their first encounter, Vox commented, projecting a holographic summary of the initial contact report. Impressed. Zarakil's translator module carried a tone of skepticism. By what? Their ability to survive without comprehensive genetic modification. The holographic display flickered, showing images of human representatives. They were remarkably symmetrical creatures, with bipedal locomotion and a complex neural network contained within a relatively fragile-looking cranial structure. Their skin varied in pigmentation, a biological adaptation that fascinated xenobiologists, a direct response to their planet's varied environmental conditions. Zarakil began the initial scanning process, her advanced equipment humming to life. Quantum-level sensors began breaking down the cellular structure of the human samples, creating multi-dimensional maps of their biological composition. Each layer revealed what seemed to be an unremarkable biological system: simple cellular structures, basic protein configurations, genetic sequences that appeared almost primitive. Standard biological preservation protocol, she muttered to herself, initiating the first phase of comprehensive analysis. The samples would be subjected to a battery of tests that had revealed the secrets of thousands of species across the galaxy. Radiation tolerance, cellular regeneration capacity, genetic plasticity, these would provide a complete profile. Her research station was among the most advanced in the Galactic Consortium. Multiple parallel processing units could simultaneously analyze biological samples from dozens of perspectives. Zarakil had personally designed many of the analytical protocols used by research stations across 7 star systems. As the initial scans progressed, Vox monitored the peripheral systems. "Shall I prepare the standard comparative databases?" The young technician knew the routine. Each new species was measured against established biological benchmarks. "Absolutely," Zarakil responded, "though I doubt we'll find anything particularly remarkable. Another standard classification seems inevitable." Little did she know how profoundly wrong that assumption would prove to be. The first set of preliminary results began to compile. Cellular density markers, genetic sequence alignments, metabolic potential indicators. Each data point was meticulously recorded. Zarakil's trained eye swept across the initial readouts, her neural implants cross-referencing the data with millennia of xenobiological research. Something was different, but she dismissed the thought almost as quickly as it formed. Humans were just another developing species, another entry in the vast catalog of biological diversity. Their time would come, But for now, they were nothing more than an interesting footnote in the grand tapestry of galactic civilization. The samples continued to process, their secrets waiting to be unveiled. Zarakil leaned back, confident in her expertise, unaware that these seemingly unremarkable biological specimens were about to challenge everything she thought she knew about life itself. In the vast, quiet expanse of the research station, the first whispers of a scientific revolution were beginning to take shape. The research station's diagnostic chamber hummed with a soft, persistent vibration. Dr. Zarakil stood motionless, her neural interfaces processing the latest batch of human biological data. What had begun as a routine analysis was slowly transforming into something far more intriguing. Vox approached cautiously, aware of the subtle change in his supervisor's demeanor. "The secondary analysis is complete," he announced, projecting a holographic display of complex genetic mappings. Zarakil's neural implants worked overtime, cross-referencing the data against thousands of known biological profiles. The human samples defied simple categorization. Where most species showed predictable genetic patterns, these samples presented a complexity that was both frustrating and fascinating. "Pull up the cellular regeneration metrics," she instructed, her voice carrying a hint of professional curiosity that bordered on excitement. The holographic display shifted, revealing intricate cellular structures that seemed to pulse with an unexpected vitality. Most species showed linear regeneration patterns, predictable, methodical, with clear limitations. These human samples demonstrated something entirely different: adaptive cellular reconstruction that seemed to almost anticipate potential damage. In the standard diagnostic chamber, a series of controlled environmental stressors were applied to the samples. Typically, biological specimens would show gradual degradation under such conditions. The human samples, however, began to exhibit remarkable defensive responses. Cellular membranes would subtly reconfigure, molecular structures would realign, creating defensive patterns that seemed almost intelligent. "This is impossible," Zarakil muttered, her neural implants running multiple concurrent analyses. Vox watched the data streams, his own neural links struggling to process the anomalies. "The cellular adaptation rates are unprecedented," he observed. Where most species would require generations of evolutionary pressure to develop such capabilities, these human samples showed immediate, localized adaptive responses. The research station's most advanced diagnostic tools were designed to break down biological systems to their most fundamental components. Yet these human samples seemed to resist such complete deconstruction. Each time the analysis protocols approached a comprehensive breakdown, the cellular structures would subtly shift, revealing new layers of complexity. Zarakil isolated a specific genetic sequence that particularly intrigued her. What appeared to be dormant genetic material in most species seemed to be something entirely different in these human samples. These weren't just inactive genetic segments, they were potential adaptive mechanisms lying in wait, ready to activate under specific conditions. "Run a quantum-level resonance scan," she instructed Vox. "I want to see how these genetic sequences respond to minimal environmental stimuli." The quantum scanner was typically used for the most advanced biological research. It could detect molecular changes at scales almost beyond comprehension. As the scan initiated, the human samples began to display behaviors that challenged everything Zarakil understood about biological systems. Minimal temperature variations triggered subtle genetic reconfigurations. Slight electromagnetic fluctuations caused cellular structures to develop protective membrane variations. It was as if the very genetic code was a living, breathing system of potential and adaptation. "The genetic plasticity is off our standard measurement scales," Vox reported, his voice a mixture of professional detachment and genuine wonder. Zarakil's research had explored biological systems across 7 star systems. She had documented species that could survive in methane atmospheres, organisms that could exist in the radiation-intense environments of neutron stars. But these human samples were different. They didn't just survive. They anticipated, they prepared, they transformed. Hours passed in the research chamber. Multiple diagnostic protocols were initiated, each revealing new layers of complexity. The human biological samples seemed to be a system of perpetual potential, with adaptive mechanisms that went beyond simple survival. "We need to expand our analysis parameters," Zorakil announced, her professional skepticism slowly giving way to genuine scientific curiosity. The samples that had initially seemed so unremarkable were proving to be anything but ordinary. As the diagnostic systems continued their relentless investigation, something remarkable was becoming clear. These human biological samples were not just another entry in the galactic catalog of life. They represented something entirely new. A biological system of such complexity and potential that it challenged fundamental understanding of evolutionary adaptation. The research station's quiet corridors seemed to hold their breath, waiting for the next revelation. In the vast expanse of scientific discovery, the first whispers of a revolutionary understanding were taking shape, one cellular adaptation at a time. The advanced research wing of the Quantum Horizon Station resembled a cathedral of scientific precision. Reinforced quantum-shielded walls contained experimental chambers designed to simulate conditions that would obliterate most known forms of life. Dr. Zorakil had spent cycles developing protocols to test the absolute limits of biological survival. Her latest project would push those limits further than ever before. The human biological samples sat innocuously in specialized containment units, their unremarkable appearance belying the mysteries they contained. Surrounding the samples was an array of diagnostic equipment so advanced it could measure cellular changes at the quantum level. "Initiate baseline diagnostic sequence," Zorakil commanded, her neural interfaces connecting with the station's primary research systems. Vox managed the primary control panel, his manipulative tendrils moving with practiced efficiency. Baseline parameters established. Preparing first test sequence. The experiment was designed to be comprehensive and ruthless. Zorakil had meticulously planned a series of environmental challenges that had systematically destroyed biological samples from over 300 documented galactic species: extreme radiation, temperature fluctuations, chemical bombardments, gravitational stress. Each test would push the human samples to their theoretical breaking point. The first phase involved intense radiation exposure. Most biological systems would rapidly break down under such conditions, their cellular structures collapsing almost instantly. Standard protocols dictated minimal exposure times to prevent complete sample destruction. But these were not standard samples. As the radiation levels increased, something extraordinary began to happen. Instead of immediate cellular collapse, the human samples began to adapt. Microscopic sensors revealed cellular structures actively reconfiguring themselves. Radiation-damaged molecular bonds were being rapidly repaired, with new protective mechanisms emerging in real time. "Increase radiation intensity to level 9, Zorakil instructed, her voice steady but tinged with growing fascination. Most research stations would have terminated the experiment long ago. But Zorakil was driven by an insatiable scientific curiosity that transcended standard research protocols. The radiation levels reached intensities that would vaporize most known life forms. Yet the human samples continued to change, to respond, to survive. Vox's sensor readings began to display patterns that defied conventional biological understanding. "The cellular reconstruction rate is exponential," he reported, his professional demeanor giving way to genuine astonishment. "They're not just surviving, they're learning from the radiation exposure." The next phase introduced extreme temperature variations, from near absolute zero to temperatures that would instantly combust most organic materials. The samples underwent a barrage of thermal stress. Each new environmental challenge seemed to trigger a profound adaptive response. Zorakil's neural implants worked overtime, processing the incoming data. What had begun as a routine investigation was transforming into something far more significant. These weren't just biological samples. They were a living, dynamic system of unprecedented complexity. Chemical bombardment followed, with molecular compounds designed to break down the most robust cellular structures. Acids, bases, exotic molecular catalysts. Each new challenge was met with an equally sophisticated biological response. The human samples didn't just resist destruction. They seemed to incorporate the very elements meant to destroy them. "This is impossible," Zorakil muttered, though her tone suggested excitement rather than genuine disbelief. Hours turned into cycles. The experimental chamber became a crucible of scientific discovery. Each test pushed the boundaries of known biological potential, and each time, the human samples demonstrated capabilities that challenged fundamental understanding of life itself. Gravitational stress tests followed, subjecting the samples to pressures that would crush molecular structures into oblivion. Yet even here, the human biological material showed remarkable resilience. Cellular structures would compress, then rapidly reconstruct, finding new configurations that maintained their fundamental integrity. As the final diagnostic sequence completed, Zorakil realized they were witnessing something unprecedented. These weren't just biological samples. They represented a form of life that was dynamic, adaptive, and possessed a complexity that defied current scientific comprehension. "Compile a preliminary report," she instructed Vox, "but keep the full extent of our findings confidential for now." The research station hummed with the quiet excitement of a scientific breakthrough. In the vast, sterile experimental chamber, the human biological samples sat quietly, their true potential only beginning to be understood. Something extraordinary was waiting to be discovered. The quantum sequencing laboratory stood as a testament to scientific precision. Massive holographic displays surrounded Dr. Zorakil, each projection revealing increasingly complex layers of human genetic material. What had begun as a routine analysis had transformed into the most significant genetic investigation of her career. Advanced molecular scanners worked tirelessly, peeling back the intricate layers of genetic information. Most species had relatively straightforward genetic structures, clear, predictable sequences that mapped out biological potential with mathematical precision. The human genetic code was anything but predictable. "Initiate deep molecular mapping," Zorakil instructed her research assistant, Vox. The laboratory hummed with anticipation. Quantum-level sensors began their meticulous investigation, revealing genetic sequences that challenged everything Zorakil understood about biological information storage. What scientists had previously dismissed as junk DNA emerged as something far more sophisticated. These weren't inactive genetic segments. They were complex adaptive networks, dormant but incredibly potent. Each seemingly random genetic sequence appeared to be a carefully designed potential solution, waiting for the right environmental trigger. Vox's neural interfaces processed the incoming data, his optical sensors widening with each new revelation. "The genetic complexity is exponential," he reported. "These aren't just genetic markers. They're adaptive algorithms." The human genetic code resembled a living, breathing system of potential. Where most species had rigid genetic structures, these samples displayed a remarkable fluidity. Genetic sequences could apparently reconfigure themselves, creating new potential pathways in response to environmental challenges. Zorakil isolated a particularly intriguing genetic segment. Under standard analysis, it appeared unremarkable, a small sequence that would typically be overlooked. But when subjected to advanced quantum scanning, the sequence revealed multiple potential activation points, each capable of triggering profound biological adaptations. "Run a comprehensive adaptive potential simulation," she commanded. The laboratory's most advanced systems came to life. Simulated environmental conditions began to interact with the genetic material, revealing layers of complexity that defied conventional understanding. Genetic sequences that appeared dormant would suddenly activate, creating protective mechanisms, adaptive responses that seemed almost intelligent in their precision. Most species had genetic codes that were essentially historical records, a blueprint of past adaptations. The human genetic material was different. It was a living system of potential, with multiple pathways always ready to activate. Hours turned into cycles of continuous investigation. Each new analysis revealed another layer of genetic complexity. The research team had long since abandoned their initial skepticism, replaced by a growing sense of scientific wonder. "These aren't just genetic sequences," Zorakil muttered, more to herself than to Vox. "They're adaptive solution generators." The implications were staggering. A single genetic sequence could potentially generate multiple biological responses, each tailored to specific environmental challenges. Where most species required generations to develop new adaptive mechanisms, these genetic structures suggested the potential for near-instantaneous biological transformation. Vox prepared a preliminary report, his neural interfaces struggling to capture the full complexity of their discovery. "The adaptive potential is beyond our current measurement capabilities," he noted. "We're observing a genetic system that appears to anticipate challenges before they occur." Zorakil recognized they were witnessing something unprecedented in galactic biological research. These weren't just genetic samples. They represented a fundamentally different approach to biological potential, a living, dynamic system of genetic information that challenged everything known about evolutionary development. The quantum sequencing displays continued their mesmerizing dance of molecular information. Each projection revealed new layers of complexity, new potential pathways of biological adaptation. Something extraordinary was taking shape in the quiet, sterile laboratory. A genetic mystery was unfolding, one that would challenge the very understanding of life itself. As the final diagnostic sequence completed, Zorakil knew their discovery would reshape scientific understanding. The human genetic code was not just a blueprint. It was a living, adaptive system of unprecedented potential. The research station hummed with the quiet excitement of a scientific breakthrough waiting to be understood. The interdisciplinary research wing of the Quantum Horizon Station buzzed with an electricity of scientific anticipation. Dr. Zorakil stood before a panel of senior researchers, her preliminary findings projected across holographic displays that seemed to pulse with living information. Immunological analysis had become the focal point of their investigation. What had begun as a routine biological study had transformed into something far more significant, a potential revolution in medical understanding that could reshape galactic medical practices. "The human immune system is not just a defensive mechanism," Zorakil explained, her neural interfaces highlighting complex molecular interactions. "It's an adaptive intelligence system that operates beyond any previously understood biological paradigm. Most known species developed immune responses through generational adaptation. A pathogen would challenge a biological system, and over multiple generations, genetic variations would emerge to provide protection. The human immune system demolished this understanding completely." Vox managed the primary data projection, his manipulative tendrils moving with practiced precision. Each holographic display revealed layers of immunological complexity that defied conventional scientific understanding. The research team watched in collective amazement as the human biological samples demonstrated an unprecedented ability to generate custom molecular defenses. Where most species would require extensive time to develop antibodies, these samples could create targeted molecular responses almost instantaneously. "Observe the molecular reconstruction sequence," Zorakil instructed, zooming into a microscopic view of cellular interactions. Pathogenic challenges that would devastate most biological systems were met with remarkable efficiency. The human immune response didn't just defend. It learned, adapted, and transformed with each new challenge. Molecular structures would reconfigure, creating defense mechanisms that seemed to anticipate potential threats before they could fully manifest. Most galactic medical systems relied on extensive genetic modification and external intervention to enhance biological defense capabilities. The human sample suggested an entirely different approach, a living, dynamic immune system that was itself a complex problem-solving mechanism. "The speed of adaptive response is extraordinary," noted Dr. Krell, a senior immunological researcher from the Galactic Medical Consortium. His neural implants were processing the data with increasing excitement. "These aren't just immune responses. They're adaptive intelligence systems." Each test revealed new layers of complexity. Viral structures that would typically overwhelm biological systems were not just resisted but systematically dismantled. The human immune response appeared to create custom molecular tools, breaking down pathogens with a precision that suggested something beyond traditional biological defense mechanisms. Zerakil isolated a specific molecular sequence that particularly intrigued her. What appeared to be a simple genetic marker was actually a complex adaptive mechanism. The sequence could apparently generate multiple defensive responses, each tailored to specific pathogenic challenges. "We're observing a biological system that doesn't just respond to threats," she explained to her colleagues. "It anticipates, learns, and transforms with each interaction." The implications were staggering. Traditional medical interventions could be completely reimagined. Instead of extensive genetic modifications or external medical treatments, this approach suggested the potential for biological systems that could heal and defend themselves with unprecedented efficiency. Hours of continuous analysis revealed increasingly complex layers of immunological potential. The human biological samples weren't just surviving. They were demonstrating a form of molecular intelligence that challenged fundamental understanding of biological defense mechanisms. Vox prepared a comprehensive report, his neural interfaces struggling to capture the full complexity of their discovery. "The adaptive potential extends beyond simple immune response," he noted. "We're observing a comprehensive biological problem-solving system." As the final diagnostic sequence is completed, the research team recognized they were witnessing something extraordinary. These weren't just biological samples. They represented a fundamentally different approach to biological potential, a living, dynamic system of unprecedented complexity. The research station hummed with the quiet excitement of a scientific breakthrough that could reshape medical understanding across multiple civilizations. Something remarkable was taking shape in the quiet, sterile laboratory. A medical revolution was unfolding, one that would challenge everything known about biological defense and adaptation. The Galactic Scientific Symposium represented the pinnacle of research presentation in 7 star systems. Holographic amphitheaters stretched across multiple dimensional planes, hosting the most brilliant minds from countless civilizations. Dr. Zerakil stood at the central podium, her neural interfaces connected to the most advanced presentation systems ever conceived. Her preliminary research reports had already created considerable buzz. Rumors of unprecedented biological discoveries had spread through scientific communication networks, generating a mixture of skepticism and anticipation. The chamber fell silent as she began her presentation. "Colleagues," Zerakil began, her voice steady and professional. "What I am about to share will fundamentally challenge our understanding of biological potential." Holographic displays materialized around her, revealing the intricate details of human biological samples. Complex molecular structures danced in 3-dimensional projections, showing adaptive mechanisms that defied conventional scientific understanding. Most research presentations followed predictable patterns: introduce findings, present data, draw measured conclusions. Zerakil's presentation was different. Each projection revealed layers of complexity that seemed to grow more extraordinary with each passing moment. Senior researchers from across the galaxy watched with increasing fascination. The human biological samples challenged everything they understood about evolutionary development, genetic potential, and adaptive capabilities. "Our initial approach was one of academic dismissal," she continued. "We viewed these samples as primitive, unremarkable. What we discovered was a biological system of unprecedented complexity." The holographic displays shifted, showing the remarkable adaptive responses observed during extreme condition experiments. Radiation challenges that would obliterate most known life forms became moments of cellular reconstruction. Temperature variations triggered adaptive genetic reconfigurations that seemed to anticipate potential threats. Dr. Krell, a senior researcher from the Quantum Genetic Consortium, leaned forward. His neural implants were processing the incoming data with increasing excitement. The implications were staggering. Zorakil's presentation systematically dismantled decades of established scientific understanding. The human immune system wasn't just a defensive mechanism. It was an adaptive intelligence system that operated beyond any previously understood biological paradigm. Traditional models suggest biological adaptation occurs over generations, she explained. These samples demonstrate adaptive capabilities that occur in real time, at the molecular level. The research team had prepared extensively for potential criticism. Scientific conferences were notorious for challenging new discoveries, especially those that challenged fundamental understanding. But the data was irrefutable. Molecular sequences that had been dismissed as junk DNA emerged as complex adaptive networks. Genetic structures displayed a fluidity and potential that suggested an entirely new approach to understanding biological development. As the presentation progressed, the initial skepticism in the chamber began to transform. Senior researchers exchanged glances, their neural interfaces working overtime to process the implications of these discoveries. "We are not just observing a new species," Zorakil concluded. "We are witnessing a fundamentally different approach to biological potential." The question and answer session that followed was intense. Researchers from multiple civilizations sought to understand, challenge, and comprehend the extraordinary findings. Each question was met with precise scientific documentation, each challenge answered with layers of complex molecular evidence. Dr. Krell was the first to speak. "These findings suggest we must completely reimagine our understanding of biological adaptation," he announced, his professional demeanor giving way to genuine scientific excitement. Zorakil's professional reputation, built over cycles of meticulous research, now stood at a pivotal moment. What had begun as a routine investigation had transformed into a potential scientific revolution. As the symposium concluded, she knew something extraordinary had occurred. The human biological samples had done more than challenge scientific understanding. They had opened an entirely new avenue of biological research. The quiet murmur of excited scientific discussion filled the holographic amphitheater. Something remarkable was taking shape. A new understanding of life itself was emerging. The aftermath of Dr. Zorakil's groundbreaking presentation rippled through multiple layers of galactic scientific and diplomatic communities. What had begun as a routine biological investigation had transformed into a discovery that would reshape understanding across entire civilizations. The Interstellar Research Consortium's central hub became a nexus of intense discussion. Representatives from hundreds of advanced species gathered in multi-dimensional conference chambers, their neural interfaces buzzing with the implications of the human biological research. "These findings are not just a scientific curiosity," announced Director Vorin, a senior researcher with millennia of scientific experience. "They represent a fundamental reimagining of biological potential." Military strategists were the first to recognize the profound implications. Species that had developed sophisticated genetic modification techniques now found themselves confronting a biological system that seemed to render their most advanced technologies almost primitive. The human biological samples suggested capabilities that went far beyond traditional understanding of adaptation. Biological defense mechanisms that could instantaneously respond to environmental challenges, genetic structures that could reconfigure themselves in real time. These were not just theoretical possibilities, but documented scientific realities. Research teams began exploring potential applications. Medical researchers saw the possibility of revolutionary treatment approaches. Where most species relied on extensive external interventions, the human biological model suggested the potential for self-healing, adaptive biological systems. Diplomatic missions that had initially viewed humans as a developing species now approached them with newfound respect. The biological research suggested a level of potential that transcended technological development. "We are not just looking at a species," Dr. Krell noted during a follow-up research symposium. "We are observing a living system of unprecedented adaptive potential." Xenobiological research centers across multiple star systems began requesting access to the original human biological samples. The demand for further investigation was unprecedented. What had once been considered a routine biological collection had become the most sought-after research material in recent galactic history. The practical applications seemed limitless. Planetary colonization teams saw potential solutions to complex biological adaptation challenges. Medical researchers envisioned treatment approaches that could revolutionize healthcare across multiple species. Zarakel found herself at the center of this scientific storm. Her meticulous research had opened an entirely new avenue of biological understanding. Researchers from hundreds of civilizations sought her insights, her detailed documentation becoming a cornerstone of a new scientific paradigm. The human biological system challenges everything we understand about evolutionary development, she explained during a comprehensive research briefing. It's not just about survival. It's about continuous, real-time adaptation. Military strategic planning underwent significant reconsideration. Species that had developed sophisticated genetic enhancement technologies now recognized that the human biological model suggested an entirely different approach to biological potential. The research had implications that extended far beyond immediate scientific understanding. It suggested a fundamentally different approach to life itself, a biological system that was not just reactive, but proactively adaptive. Collaborative research initiatives began to form. Species that had previously maintained strict scientific isolation now sought collaborative opportunities to further investigate the human biological discoveries. We are witnessing the emergence of a new understanding of biological potential, Director Voren announced during a comprehensive galactic research symposium. These findings will reshape our approach to biological sciences for generations to come. The human biological samples had become more than a scientific discovery. They represented a window into a new understanding of life's potential, a living testament to the extraordinary complexity of biological adaptation. As research continued, the initial excitement began to transform into a more profound scientific appreciation. What had seemed impossible was now documented scientific fact. The human biological system was not just unique, it was revolutionary. The quiet research stations hummed with the excitement of a scientific breakthrough that would echo across generations. Something extraordinary was taking shape. A new horizon of biological understanding was emerging. In the vast, complex tapestry of galactic scientific exploration, the human biological discovery stood as a beacon of unprecedented potential. The research archives of the Quantum Horizon Station stood as a testament to scientific exploration. Dr. Zarakel sat in her personal reflection chamber, surrounded by holographic projections of the human biological research that had transformed her entire scientific understanding. Cycles had passed since that first mundane assignment. What had begun as a routine investigation had become the most significant scientific discovery of her career, perhaps of multiple generations. Her neural interfaces displayed a comprehensive timeline of their research. Each projection told a story of transformation, from initial skepticism to profound scientific wonder. The human biological samples had done more than challenge scientific understanding. They had reshaped her entire perspective on life itself. Vox, she called to her research assistant, compile the final comprehensive report. The chamber hummed with the quiet energy of scientific reflection. Zarakel reviewed the journey that had brought them to this moment. The initial diplomatic samples that had seemed so unremarkable now represented a fundamental shift in understanding biological potential. Her earliest notes were filled with professional dismissal. Words like primitive and unsophisticated stood in stark contrast to the revolutionary findings that followed. Each stage of research had peeled back another layer of complexity, revealing a biological system of unprecedented adaptive potential. The extreme condition experiments played across one holographic display. Radiation challenges that would obliterate most known life forms became moments of cellular reconstruction. Temperature variations triggered adaptive genetic reconfigurations that seemed to anticipate potential threats. Another projection showed the intricate immune response mechanisms. What had initially appeared to be a simple defensive system was actually a complex, intelligent adaptive network. The human biological samples demonstrated an ability to learn, transform, and respond with a sophistication that challenged fundamental understanding of life itself. Zarakel's professional reputation had been forever changed. The scientific community that once viewed her with professional skepticism now sought her insights. Her research had opened an entirely new avenue of biological understanding. The human biological system is not just a collection of cells, she dictated for the final report. It is a living, dynamic system of extraordinary potential. The diplomatic implications played across another display. Species that had viewed humans as a developing civilization now approached them with newfound respect. The biological research suggested a level of potential that transcended technological development. Vox prepared the final documentation, his neural interfaces carefully compiling years of research into a comprehensive scientific narrative. Each data point, each experimental result told a story of discovery that would reshape scientific understanding for generations. Personal transformation accompanied the scientific journey. Zarakel reflected on her initial approach, the professional dismissal, the academic arrogance that had initially colored her research. The human biological samples had taught her something profound about scientific exploration. True discovery requires humility, openness, and a willingness to challenge existing understanding. The final report was more than a scientific document. It was a testament to the extraordinary potential of scientific exploration. Each page revealed layers of complexity that suggested an entirely new approach to understanding life itself. Submission ready, Vox announced, the final holographic projections crystallizing into a comprehensive research document. Zarakel's groundbreaking paper would be titled Humanity, a Biological Enigma, a title that captured the essence of their extraordinary discovery. It represented the culmination of cycles of research, a journey from professional skepticism to profound scientific wonder. As the final documentation compiled, she understood that their research was more than a scientific breakthrough. It was a reminder of the extraordinary potential that could be discovered when scientific exploration was approached with genuine curiosity and an open mind. The research station hummed with the quiet excitement of a scientific journey completed. Something remarkable had taken shape. A new understanding of life's potential was emerging. In the vast, complex tapestry of galactic scientific exploration, the human biological discovery stood as a beacon of unprecedented potential, a testament to the extraordinary possibilities that await those willing to look beyond established understanding.